Good day, colleagues, and welcome to our second Hannover and Lancet Laboratories webinar. My name is Santi Lesitlaba, Marketing Manager at Lancet Laboratories, and it is my great pleasure to welcome you all. Thank you for taking the time at this latter part of the year. As introduced, this is our second webinar that we are having this year, and we felt the need to have the second one towards the end of the year to keep you all up to speed in terms of all developments in our space, in the life insurance industry space and the laboratory space. So we're really thankful that all of you could make it today. Some of you are joining us from the first webinar that we had in the year, and some of you, it might be the only first time that you're joining us, but welcome to you all. I don't want to take up too much time because I think we've got a very action-packed and interesting presentations from our subject matter experts today. But I just want to share a history to all of you in terms of the collaboration that is shared between Lancet Laboratories and Hannover Re. I think the collaboration spans back to about 6 years ago. And I think it was an unexpected collaboration, firstly from a Hannover Re, who is a leader in their space in terms of reinsurance, and Lancet Laboratories, who is also a leader in terms of the diagnostic service provider space. I think the collaboration was spurred on a need to ensure that the industry as a whole, the respective industries, get a lot of insight, education, awareness, and the relationship started off with physical seminars. But because of the advent of COVID nationally and internationally, we felt that we couldn't stop this opportunity to have our colleagues share the knowledge and information that was in our hands. So we started these webinars from 2020, and we're quite pleased that they were a success from the first one to the second one that you have joined us all on. And we're planning to continue in the same stride, offering these webinars into the new year. I think the relationship has got also a lot of interesting collaborations in terms of data sharing, but I don't want to give too much away. I think in 2021, Hannover Re, our partners, will be in a position to share the interesting developments that we have collaborated on, that will also be cutting edge and will allow the industry, the life insurance industry and the laboratory space, to share some valuable outcomes for our customers that we all share.
Our next speaker is Dr. Liana Maria. She's a clinical virologist with Lancet Labs, and she's originally from Pretoria, where she obtained her medical degree. After doing her internship in community service, she then started training as a virologist, which she obtained her specialization as a virologist cum laude at the University of Stellenbosch, and she has a special interest in viral lower respiratory tract infections in children. During her postgraduate training, she also obtained a diploma in tropical medicine and hygiene from WITS, and interestingly enough, which must have been quite an interesting time for her, for 18 months she was actually the only clinical virologist in the entire Eastern Cape. And I think given what we've seen occurring in the Eastern Cape in terms of the pandemic and the strain that health system is under, I suppose it's sad, but I suppose not unsurprising that was actually the situation. So she's been with Lancet since 2015, and I'm sure that you can all agree then that we've got the right person for our next talk, which is going to be discussing the very topical issue of vaccines. So Dr. Marie, I will hand over to you.
Hi, Matthew. Thank you so much for that introduction. Yes, I must say I am quite sad to hear what is going on in Port Elizabeth at the moment. Let me quickly share my screen. Okay, I hope everyone can see my screen because it's, I can see it, but I'm not sure if you can. So is it right?
Yeah, it's right.
Okay, perfect. So this is hopefully not too long update on the vaccines, and everything changes. Like I literally saw an update last night while I was waiting for President Ramaphosa to speak, so it may have changed by now again. So this is just to say why we urgently need our COVID vaccine. So unfortunately, worldwide, every population is still at risk. If you look at the graph, I've just included sort of some of the top countries in South Africa to show that, yes, indeed, we have started our second peak. So if one looks at serosurveys, there are still large immunity gaps. There was a big meta-analysis where the seroprevalence estimate was 3.38%, and it ranged from 0.3% in Malaysia to 22% in Iran. That meta-analysis only included one study from Africa, from Kenya, where the prevalence was 5.6%. Published, I've only been able to find that one study from the Cape Town metro area where they did seroprevalence surveys in antenatal and HIV clinics, where their seroprevalence was 40%. I can just say anecdotally from the samples we have received so far for antibody testing, but that is pre-selected, so it's not generalizable, but we are around 20 to 24% seroprevalence from the samples we receive. Then, of course, we're all stuck in COVID fatigue at the moment, where strict social distancing and lockdown restrictions are not sustainable, and we all want to see our friends and families for Christmas. And of course, none of us can go at it alone. So this is just a little slide to show how immunity develops. So the important bits on the left, sorry, let me get my pointer out. Here we go. So here we see the spike protein is the important bit of the virus because that's on the outside of the virus, and that is the protein that locks onto the ACE2 receptors to enter the cell. And there's one specific area of the spike protein called the receptor binding domain that from the name binds to the receptor, and that seems to be the main target for neutralizing antibodies. So on the right, we can see our immune response is, first you have your helper T cells, so they will, first you have your antigen presenting cells. So they engulf the virus and break it up and then present the antigens of the virus to the helper T cells, and then the helper T cells then help or promote either your B cells or your T cells to attack the virus. So B cells will make antibodies and your T cells will just release cytotoxins and granzymes and things to kill the virus. Sorry. Okay, sorry. There we go. Sorry. Laser pointer and going to the next slide seems to be difficult. So the different types of vaccines is just a little bit of an introduction. So basically we've got three types: the live attenuated or the inactivated vaccines, the protein subunits or peptides, and then where viruses are involved. So it's either a virus vector or DNA or RNA. Okay, so our inactivated and live attenuated virus vaccines. So with inactivated vaccines, the viruses are grown in culture, but then they need to be inactivated or killed to be safe. But during the killing process, the epitope, so that's the spike proteins on the outside of the virus, you see here, they need to remain intact so that your immune system can actually recognize them and make B cells and T cells against them. So the potential problem of this is during the denature or this killing process, you can denature these proteins, you can degrade them, they can stick together. So those are sort of all of the problems or potential problems with an inactivated vaccine, but they are a cost-effective way to make a vaccine. The ways to inactivate the viruses are formalin and ultraviolet are 2 examples, but you also need your biosafety level 3 laboratory to develop these for the SARS-CoV-2. Then your live attenuated vaccines work well for acute infections or acute diseases like the MMR for measles, mumps and rubella, or the varicella vaccine for chickenpox. But we are potentially worried about safety and efficacy issues with a live attenuated virus for chronic infections, especially HIV. The good thing for a live attenuated vaccine is that it induces immune responses against several antigens, so it's not one that is cherry-picked, but it can be against the spike protein or a different part of the spike protein, or from the nucleocapsid, sort of the membrane around the virus. There is a possibility for mass scale-up production. The big problem we're always worried about is then recombination, especially with SARS-CoV-2, to recombine with a wild-type coronavirus, one of our seasonal ones, for example, and then maybe they'll be, I don't know, more easily admissible and spreadable, or cause more severe disease if it gets one of the other mutations or from SARS-CoV-1, for example, if SARS-CoV-1 ever comes back out again. And the big problem in South African context is that all live attenuated vaccines are contraindicated in immunocompromised patients because that is the one group where we're the most worried about getting a severe reaction to the virus because you can't control it. Then the protein subunits or peptide vaccines. So these, you only use the specific antigen fragments from the virus that you know is the main targets for your immune response. So that's where most of them I'm going to discuss includes the spike protein. So these types of vaccines eliminates concerns of incomplete inactivation, virulence recovery with the live attenuated, or with a vaccine vector vaccine, you're always worried about pre-existing immunity against the vector. So none of these are of issue or are an issue with a subunit vaccine. You can specifically target well-characterized neutralizing epitopes. So the epitopes is the protein or peptide that you know your immune system will recognize as foreign and then mount a response against it. Oftentimes it's used in combination with adjuvants to improve the immunogenicity and efficacy of the vaccine. And as I've mentioned, most of them target the spike or S protein. The peptide vaccines, the peptides are just sort of smaller amino acid sequences than proteins. So they're less well defined exactly what they can do. They don't really obtain like a secondary or tertiary structure. But they've most of the problems with subunit vaccines, they will also have pure immunogen, so you need your adjuvant to enhance immunogenicity. Okay, sorry, there we go. Then viral vector vaccines. So we either have non-replicating viral vector vaccines or replicating viral vectors. So the non-replicating vector, as the name says, it is dead, it won't replicate in your body. The replicating one will. So gene of the coronavirus, or the spike gene, is inserted into the vector, and then the vector is delivered to the body, whether that is due with an injection or anything like that. And then the virus needs to enter your cells, and then the proteins are translated, or sorry, the gene is translated, and then the proteins are expressed and immune system goes. So the potential problems with this is one, potentially you're worried about the spread of the vector virus. If the vector virus spreads extensively in your body, so of course it's a systemic infection that your immune system can't get a hold of. That is one of the things people are potentially worried about. On the other side, one is also worried about duration of the expression of the gene or of the protein. Is the protein expressed in sufficient quantities that you can actually mount a response against it? So a lot of the times, the administration of the virus vector, you have to give a lot because they're not replicating. People are also potentially worried about a chromosomal integration, but if it's a RNA-based viral vector that does not have a reverse transcriptase enzyme, it cannot integrate into your DNA at all. And the big problem, as I've mentioned previously, is the pre-existing immunity against the vector. So if you already have immunity against the vector and you then inject it into a person, your immune system can attack it before it can actually produce the protein for your immune system to recognize. And that can render the vaccine ineffective. So a lot of the time with a viral vector-based vaccine, there will be a prime boost strategy where you give the vector either usually as the prime and not the boost, and then you boost, or the other way around, you prime it with a non-viral DNA virus and then boost with the vector, or two different vectors. I will mention an example of that soon. Then we've got our nucleic acid vaccines, so that's DNA-based or RNA-based. So with DNA-based ones, it contains the viral gene in the form of a DNA plasmid, and that is then delivered either by intramuscular injection of the naked plasmid, electroporation, gene gun, they use a lot of terminology, and then so the vector needs to be then transported into the cell nucleus to function. That is the big difference between the DNA-based and the RNA-based vaccines, because DNA-based needs to work where your DNA is, so it can use your enzymes, and that is in the nucleus. So with a DNA-based vaccine, you a lot of the time have to give that polysaccharide adjuvant to enhance your immunogenicity of the vaccine. None of these DNA-based vaccines have been approved to date, but clinical trials have shown at least that there is no chromosomal integration of the plasmid DNA, nor development of anti-DNA antibodies. So that worry or challenge doesn't seem to be an issue here. And as I've mentioned, it's part of a prime boost strategy with a viral vector-based vaccine. So, sorry. The next one is our RNA vaccine. So these are a lot of the ones I will discuss now are based on RNA vaccines. So they only contain the selected viral genes in the form of messenger RNA. So messenger RNA is the RNA bit that is directly translated into a protein. But around the RNA, there has to be a lipid coat, and that helps to enhance cellular uptake. And then also, importantly, it helps to keep the RNA for longer and prevents it to degrade. So the mRNA is highly sensitive to degradation because it is single-stranded structure, and your body contains RNases those are the enzymes against RNA, to degrade them because you don't want foreign RNA in your system for long. And of course, no additional mRNA copies are made. So you have to give a sufficient dose of mRNA for you to translate into proteins to present to your immune system to make antibodies and T cells against. So to improve the stability of the messenger RNA, they are placed in that lipid nanoparticle. The companies also look at changing some of the nucleotides to make them more stable, and also a process which they call sequence optimization, just to make sure, just sort of, sometimes they add bits at the end or in the beginning of the sequence that helps to stabilize it, or if it's cut off, it is nonsense that is cut off and not actually the real gene. So the other interesting thing some manufacturers are looking into is what they call self-amplifying RNA. So that is where they use the genomic backbone of that contains the viral replication machinery of certain viruses that actually have all of the genes and enzymes they need to self-replicate. They don't need your host cell machinery, they bring what they need with them. So they are able to generate their own messenger RNA. So you have an improved immune response and you require smaller doses. So this is just to show about the different clinical vaccine testing and approval. So we've got preclinical, human testing, and then post-clinical. So preclinical is in vitro, so it's in cells and cell lines, and in animals, you try to establish the appropriate dosage, look at pharmacokinetics and toxicology data. You don't want to give too little, you don't want to give too much. Then human testing, phase 1, we look at vaccine safety, we find the correct dosage in humans, and to make sure that people are actually able to produce an immune response against the virus, and that is usually done in a small number of healthy subjects. Then in phase 2, you evaluate initial safety and efficacy data in a larger group, is usually a few hundred people, and then in phase 3 trials, you have more definitive evidence of efficacy, so it's large randomized blinded trials, which includes hundreds to thousands of people. And then post-approval safety monitoring is what happens with all vaccines after they've been approved, that if anyone develops any side effects or complications after the vaccine, it is reported to the different bodies so that they keep track on exactly what is happening, to see if there is a very rare side effect that happens, like in 1 in 50 million people, that they pick it up through that way. So this is just a graphical representation to show this process. So it takes years and years and years, about 10 to 15 years. So in this slide, we can see for some of the vaccines, it takes even longer than that. Rotavirus took 33 years, polio vaccine took 47 years, measles seems to be one that was a lot shorter, but now with COVID, we are so worried about it. They have this accelerated process of vaccine development. So all of the phases are started in short succession after each other, and they overlap. So in the next few slides, when I speak about specific vaccines, a lot of them I have interim analysis, or I have, yes, there is a phase 2 trial, but they have not published anything. So, but they are including all of the necessary ideas. But they're also bypassing and shortening all of the bureaucratic hurdles, which have little or nothing to do with actual safety and efficacy. So that has dramatically shortened the process to go from 10 years to maximum 2 years, or now we even have vaccines that are available within 10 months. So if we now look at the COVID-19 vaccine specifically, this is the one I had to update in the last week or so to show, yes, we have one vaccine, the Fisa vaccine, that has been approved in the UK, Canada, and in the US. They are speculating in the US that they should approve the Moderna vaccine by the end of the week. Okay, so if we start with Moderna first, it is a lipid nanoparticle encapsulated mRNA vaccine that encodes the stabilized pre-fusion SARS-CoV-2 spike protein. So in the phase 1 study, so that was, as I say, phase 1, small amount of participants, only 45 adults. They looked at 3 doses, and all participants had an immune response to the vaccine. The adverse events were not severe, mild and moderate only: fatigue, chills, headache, myalgia, pain at the injection site. There were no serious adverse events. They decided to, the one I've highlighted in red now, the 100 micrograms vaccine, that is the one they decided to take forward into phase 2 and phase 3 trials. And in that group, the antibody levels has remained elevated up to day 119. So just to show on the graph, you can see the last line in this graph and in subsequent graphs is always where they look at this antibody responses in convalescent samples from patients who had COVID-19 and have survived, to see whether the immune response after the vaccine is as good as what we see in patients who survived natural infection. So the phase 2 trial, they wanted to confirm the dose. They included 600 participants. They mentioned that they still use a 50 microgram group, but they have not posted any data, so I cannot say exactly what they found there. Then the big one is with phase 3 trial. So this was a randomized 1-to-1 placebo-controlled study. They include 30,000 participants older than 18 years of age in the US. More than 7,000 of them were older than 65 years. More than 5,000 participants in the lower age category had comorbidities, and they received the 100 microgram dose, so 2 doses 1 month apart. The primary endpoint they looked at was prevention of symptomatic COVID-19, and key secondary endpoints were prevention of severe disease and prevention of SARS-CoV-2, of infection by SARS-CoV-2. So these were their results. So they had 196 cases of COVID-19, 185, the majority of them were in the placebo group, only 11 in the vaccine group. So that gives an efficacy against COVID-19 of 94%. Once again, the adverse reactions were pain at injection site, fatigue, myalgia, headache, and also redness at the infection site. Then they only had 30 cases of severe COVID-19. All of them were in the placebo group, so they felt that efficacy, so from that data, efficacy against severe COVID-19 was 100%, and there was only 1 COVID-19 related death, and that occurred in the placebo group. And in the study also, I just needed to mention the participants. It was in the US, but they included a lot of different ethnicities, so it wasn't just one ethnic group that they included in their study. So this is the Pfizer Biontech vaccine. So this phase 1, they once again had a placebo-controlled dose escalation study. So they had 2 candidate vaccines. I'm just going to call them B1 and then B2. And they gave 2 doses 21 days apart. So B1 was also, like Moderna, a lipid nanoparticle encapsulated nucleoside modified mRNA, but it only included the receptor binding domain that was trimerized, so they made 3 copies that stuck together to increase reactogenicity. B2, very similar to Moderna, had the stabilized pre-fusion spike protein. Included only 195 participants, all of them were older than 18 months of age. And this, I'm highlighting again the one they've chosen. They chose the B2 vaccine, the 30 milligram dosing, and we can see once again the immune responses were similar to the convalescent samples also tested. And they chose B2 to take forward due to its lower incidence of and severity of systemic reactions. So basically the same types of local reactions that we saw with Moderna, more frequent after the 2nd dose. They were quite mild and they usually peaked about day 2 after infection and then subsided a few days later, the side effects. So if we look at their phase 2-3 data, so they combined phase 2 and a phase 3 trial, once again placebo-controlled 1-to-1. Sorry, they included more than 40,000 participants who were older than 16 years of age in 6 countries. The US, Argentina, Brazil, South Africa, Germany, and Turkey. The majority of the participants were from the US, and the majority were white. And 42% of the participants were older than 55 years of age. So they got the 30 microgram dose 21 days apart, and their primary endpoint was prevention of confirmed COVID-19 at least 7 days after the 2nd dose, looking at local and systemic adverse events, and then secondary endpoints were prevention of severe disease. So these are their results. They had 170 cases of COVID-19, 162 in the placebo group, 8 in the vaccine group, which gave them efficacy against COVID-19 of 95%. There were 10 cases of severe COVID-19 after the 1st dose. They didn't mention how soon after that or how many were after the 2nd dose, but they said only after the 1st dose they counted from severity, and there was only 1 in the vaccine group. Once again, the same local side effects of mild to moderate pain at injection site. And interestingly enough, their side effects were less frequent in older patients. So you can see here the patients older than 55 years of age. Systemic side effects were also fatigue, headache, fever, chills. Once again, more often in the younger group and after the 2nd dose. One would assume it is in younger people because a lot of these side effects is actually also related to your immune response. Younger people will have a more robust immune response than older people. Adverse events that were noted was lymphadenopathy, and there were 4 cases of a vaccine-related serious adverse event. One was a shoulder injury after the injection. The lymphadenopathy, 1 patient had a ventricular tachycardia, but there were no COVID-19 related deaths. So as I mentioned, this is the vaccine that has been approved by the UK, by Canada, and by the FDA over the weekend, and this is the one that has been administered in the UK and in the US. So the next one is the Oxford Astrazeneca vaccine. So it's a replication-deficient chimpanzee adenovirus vector that encodes the SARS-CoV-2 spike protein. So in the phase 1/2 trial, it was a single-blinded, randomized controlled trial with just over 1,000 participants, all in the UK, all adults older than 18 years of age, and they included 3 groups. So a single dose of the vaccine, a single dose of the meningococcal meningitis vaccine. So they decided to, in the control group, not use saline or any that were used in the other trials, because they said they found with viral vector-based vaccines, a lot of people have sort of noticed the local side effects of the pain at the injection site, and fatigue and things like that more commonly after a virus vector vaccine than after the placebo. So they were worried that people would get the vaccine and have less side effects and know that they had the placebo, so they would underreport or overreport any sort of adverse events that they received in the vaccine. So they gave them a vaccine that is also known to cause sort of pain and injection site and redness to try to mask for the participants what they got. And then in a small group of about 10 patients, they used a prime boost strategy where they gave 2 doses of the vaccine 28 days apart. As I mentioned, median age of the group was 35 years and 90% of them were white. So they had local and systemic reactions were more common in the vaccine, well, the coronavirus vaccine group than in the meningitis vaccine group, and the responses were less severe after the booster dose. So it was actually then also the booster dose that they decided to take forward to the phase 2/3 trials. There were no serious adverse events in the vaccine group, and they had both T-cell immune responses and antibody immune responses after the vaccine. In this study, 2 of the sites halfway through the study allowed the patients to take prophylactic paracetamol Panado before they got the shot and to continue about 24 hours after they got them. And the prophylactic paracetamol was able to reduce a lot of the commonly reported reactions, the pain, fatigue, malaise, myalgia, that people reported after the vaccine. So in the phase 2/3 trial, so this is still interim analysis data I'm going to present now. So there are 4 ongoing blinded randomized controlled trials going on in the UK, Brazil, and South Africa. So more than 23,000 participants have been included, more than 18 years of age, but this data is only on 11,000 participants in the UK and Brazil. They haven't included any of the South African data as yet. 86% of the patients were between 18 and 55, 60% of them were female, and the majority of them were white again. So with this study was interesting. They wanted to compare the prime boost strategy with the control, and this time, depending on the sites, they either use saline or the meningitis vaccine. But at 1 or 2 sites, or I should probably say, there was a problem with the manufacturing process of the vaccine itself, because they, I'm going to say farmed it out, but they used different manufacturers to produce the vaccine. And depending on where it was done, they used different quantification methods to make sure that there was enough, virus in the vaccine. So they either use spectrophotometry or quantitative PCR. And initially they thought they were equal, so they said it's not a problem to use which one. But one of the manufacturers used the spectrophotometry, and in the end, they actually produced, even though the values were the same, they produced a lower dose vaccine. So and then later on in the process, they said no, they sort of got everything back to they have to use the quantitative PCR to determine the quantity of virus in the vaccine. So initially, the regulators were really worried that the patients who got the low-dose vaccine at these sites got an inferior vaccine, so they wanted to sort of stop the trial and they re-looked at it, but they decided to continue the trial and present their data because binding antibodies produced by participants who got the low-dose vaccine were at the same quantity or levels as the standard dose vaccine group. So they allowed the trial to go through with the results. And it's a good thing they did, because that was the really interesting part about this study, was the differences we see in the efficacy between the standard dose group and the low dose group. So there were 131 cases of symptomatic COVID-19, 30 in the vaccine group and 101 in the control group. So the overall efficacy of the vaccine was only 70%, not 94, 95 that we saw with Moderna and the Fisa vaccines. But when they broke that down, they saw that, strangely enough, the low dose vaccine, so there was a low dose prime, standard dose boost, only 3 cases of COVID-19 were seen in the vaccine group versus 30 in the control group. So that gave an efficacy of 90%. Whereas in the standard dose group, the efficacy was only 62%. So 10 participants in the study were hospitalized due to COVID-19. 2 of them were severe, 1 participant died, but all of the hospitalizations occurred in the control group. This was also the trial that was in the news because there were 3 cases of transverse myelitis during the study where they had to halt the study and investigate. So 1 case was in the control group, 2 cases were in the vaccine group. One of those cases was actually a patient with undiagnosed multiple sclerosis. So only 1 they felt was possibly related to the study the patient was involved in, and they let the study continue. So the other interesting thing about this study is that this is the only study where with this in the UK, they ask the patients not only to contact the trial if they develop symptoms to be swabbed, but they sent the patient swabs to do a weekly self-administered swab to look for asymptomatic infection. So in that specific subgroup, once again, it was the lower dose group that had showed an efficacy against asymptomatic or symptom unknown infection with SARS-CoV-2 of 58%, whereas in the standard dose group, it was only 3%. Whether this can be translated into larger numbers and not be due to an unforeseen but good thing that happened during the manufacturing process, I think they need to take forward to see exactly what is going on. And over the weekend as well, they've mentioned that Astrazeneca and the Sputnik vaccine group in Russia, I'm going to mention now, they are starting a new phase 3 trial where they're going to prime with the AstraZeneca vaccine and then boost with the Sputnik Gamaleyan Institute vaccine to see with what response they can get from using 2 different viral vectors. I'll mention that again. So just a little summary of these top 3 vaccines. So I've mentioned, so both are prime boost, 2 doses, 4 weeks apart or 3 weeks apart. The big thing for us is the storage and shelf life of the vaccines. Moderna can be kept at -20 for 6 months, fridge temperatures for 30 days, and then room temperature for 12 hours. Big one, Fisa has to be kept at -70. They do say now it is stable for 5 days in fridge temperatures. For Oxford or Astrazeneca, it is fridge temperatures for 6 months, which is a good thing. And the next thing for South Africa is the price difference. And AstraZeneca is also the only company that has said they're not loading that price until the pandemic ends. So it will remain at the lowest level. And they are also able, or they're promising at the moment, to give the highest number of doses during the next year or 2. So just have a few slides on some of the other vaccines. So that's the Johnson & Johnson vaccine. It is also an adenovirus vector. This is adenovirus 26 vector that expresses the SARS-CoV-2 spike protein. That is the same vector they are using for their Ebola vaccine that has been approved by the European Union. I couldn't find much more information about this specific vaccine. They just said a single dose induced a robust immune response, both neutralizing antibodies and T-cell. It was well tolerated. There were only 2 serious adverse events, 1 case of hypertension that was not vaccine related, and a pyrexia of unknown origin, which I thought maybe someone had COVID, but they recovered within 12 hours and they didn't get COVID. Then there is an ongoing phase 3 trial in the US, several countries in South America, Europe, and South Africa once again. They initially said they wanted to include up to 60,000 participants older than 18 years of age, but due to the massive epidemic that they're seeing in the US, they feel that only getting to about 40,000 participants in the US, they'll be able to get enough data to see what is the efficacy of this vaccine. They do mention they're still looking at a single dose or 2 doses, but I haven't seen any more information. So this is also the vaccine where Johnson & Johnson and Aspen Pharmacare have a preliminary agreement that the vaccine will be produced or packaged in South Africa in Port Elizabeth. And late last night, Johnson & Johnson also said that they have submitted their application to SAHPRA for approval of this vaccine. The next one is the Nurvivex vaccine. It's a protein, so it's once again just a spike protein. So the interesting thing about this vaccine is they use a plant virus, a baculovirus, to deliver the genetic sequence for the spike protein to full armyworm moth cells. So it's the cells from moths, and the spike protein is inserted into them. They then translated, produce the spike proteins, and the spike proteins are then studded onto a synthetic particle. And then together with a saponin to adjuvant, which they call matrix M, that is then given to participants. So once again, not much information. The phase 1 trial data showed it was well tolerated, mild reactogenicity, and all of the participants developed neutralizing antibodies after the 2nd dose. Phase 2 and 3 trials are ongoing. In the UK, it's a phase 3 trial of about 15,000 participants. In South Africa, there is a phase 2B trial ongoing, where they've included just over 4,000 participants, including 245 HIV positive patients who are medically stable with regards to their HIV. They're also planning to start a large phase 3 trial in the US and Mexico soon. And this one is also stable for weeks at 2 to 8 degrees. So a little bit about one of the Chinese vaccines, Convadecia. It's an adenovirus vector again that expresses the spike protein. Not much information on this one. Large phase 3 trials in Saudi Arabia, Russia, Pakistan, Mexico, and Chile. And they have said that they'll be able to produce a lot of vaccines. Then the Sputnik one, that's the one I've mentioned earlier. So it's an adenovirus vector that expresses also the spike protein. They prime and boost with 2 different adenoviruses, and that is to get away from your immune response to the priming adenovirus, that if you don't make antibodies, that it doesn't negatively affect your immune response to the boost. Once again, I haven't been able to find a lot of data. They say their phase 3 study is ongoing. They claim it's more than 95% efficacy at 42 days after the first dose. More than that, I really unfortunately cannot say, but this is the one where they want to combine this adenovirus 26 with the Oxford vaccine to see how that will work. So last little bits is with the vaccine doses. This is to show how many vaccine doses have been secured by different governments. We can see India has 1.6 billion, the European Union 1.4 billion, the US 1.1, and if we move on, so we can see that high-income countries have secured 3.5 billion doses, middle-income countries over 800,000 million doses, lower middle-income countries 1.75 billion, and then with low-income countries, nothing yet, and that's where the COVAX alliance will hopefully help. So to just reinforce, sort of stir it again, if you look at the United Kingdom, for example, so they have secured over 355 million doses. If you look at the population of the UK in 2019, they said it was 66.65 million people in the UK. So they have secured more than 5 times the population in the UK of vaccines that they say they want to take up. So looking at the cost of the vaccines, they go from $3 to $4 for the Astrazeneca vaccine to $37 for the Moderna vaccine. So that's from 60 Rand to 600 Rand, which in the South African situation, and that's per dose. So it's ×2 for all of them, because most of these are prime boosts. So that is the big worrying factor in our country. So if you look at the 5 key considerations in choosing the most appropriate one for South Africa, we need to start looking at safety. So is it safe in everyone, in older patients, in younger patients, in pregnant women, in immune-compromised patients, which is a big thing in South Africa? The efficacy, is the efficacy that they say in their studies, is it similar across all of the populations included in the trial? And that's why a lot of the studies now specifically say, how many people were included who had comorbidities, how many people were included from different ethnicities and races, to show that efficacy is the same across all different populations. Cost, we also need to look at in South Africa. Then ease of storage and administration. That's the big problem with the Pfizer vaccine that needs to be kept at -70. and then of course the number of doses. But at the moment it seems we won't be able to get away from 2 doses. So this is where a lot of our hopes are sitting at the moment, is with the COVAX Alliance. So that's a coalition led by the WHO, Gavi and CEPI. And the COVAX facility is designed to guarantee rapid, fair and equitable access to the vaccines for every country in the world by sharing the risk associated with the vaccine development, investing in manufacturing upfront, so the vaccines can be deployed at scale as soon as they are proven to be successful, and pooling procurement and purchasing power. So the goal is for this alliance is to secure 2 billion doses by the end of 2021. So we have only expressed an interest to be part of the facility. The problem for South Africa is that we are not seen as one of the 92 economies that will be supported by the COVAX Advanced Market Commitment. So we are seen as part of, we earn too much money, or the country earns too much money to be boosted or sponsored by the other people taking part in COVAX. So we are part of the group that needs to subsidize or cross-subsidize these other economies. So we are in the same group as New Zealand, Japan, Monaco, Switzerland, etc. So that is a problem for South Africa, that we are not poor enough to get the full benefit of COVAX, but at least as part of the larger group, we have collective bargaining power with the vaccine manufacturers. So South Africa and India have also called on the World Trade Organization to suspend intellectual property rights related to COVID-19. So not only the vaccine, but also medications, ventilators and things like that. But we will need to see if the World Trade Organization will do anything about it. The article I read sort of said no one was really interested in doing that. So if we look at vaccine availability in South Africa, initially the articles came out that said we have paid our down payment of 500 million rand to access the vaccines through the COVAX initiative. The one problem is that, and that is for enough vaccines for 10% of our population, and the full cost of this will be 2 billion rand. For the vaccines, for 10% of our population, that excludes storing the vaccine, distributing the vaccine, and administering the vaccine. So the real cost will be much higher. Then after this article came out, more articles came out to say we have not actually made that 500 million rand down payment, but the Solidarity Fund has now said that it would donate 327 million as part of that down payment. And the other base thing is, from this, the COVAX vaccine, our best case scenario is that we will only have a vaccine in the second quarter of next year. And as I've mentioned, that's only for 10% of our population. So one always needs to also look at fair vaccine allocation. So according to the fair priority model, fundamental values, one needs to look at benefit and minimizing harm, prioritizing the disadvantaged and equal moral concern. And the WHO suggests that countries should receive doses proportional to their population size, and they suggest anything from 3 to 20%. So the specific article I'm quoting here is from 2 ethicists or medical Stellenbosch, and they suggested that these should be the first group of people included in the first rollout of the vaccine in South Africa. I have not seen any sort of other guidelines locally to say who should get it first. So they include all frontline workers. If we don't get enough doses, they then suggest then healthcare workers at the highest risk. So it's frontline healthcare workers, and then high-risk frontline workers in essential services. So that does not include only doctors and nurses, but all essential services. Then people with comorbidities, which place them at increased risk of severe disease or death, and then people with other comorbidities, especially if they live in crowded circumstances. And they, just one quote from their side, rationing processes should be fair and based on transparent, consistent criteria that can be subjected to objective scrutiny with the goal of ensuring accountability, equity, and fairness. One last thing that they mentioned in their article that I think we should mention here is we don't all have COVID fatigue, but the anti-vaxxer movement is strong in South Africa. So there was a recent Ipsos survey which indicated that only 64% of South Africans would accept a vaccine once it becomes available. So not only them, but other medical professionals have said, even though the vaccine is only coming next year sometime, we need to start sending out the message now to convince or to tell people that why the vaccine is needed, and to show them that all of their fears regarding the vaccine and its administration is unfounded, because we need more than 36% of South Africans to be willing to take the vaccine. So this is just the last slide with an interesting metaphor about defending against pandemics, which they call the Swiss cheese model. And that is to show that no single intervention is perfect at preventing the spread of SARS-CoV-2. Each intervention has its own holes, but multiple layers improves success. And that's it. Any questions?
Thanks very much, Dr. Marie. I can't actually see any questions at the moment, so I don't know if there might still be one or two that come through. But while I'm talking, I'll let you know. Thank you very much. I think that was quite an in-depth view of vaccines. I know I certainly learnt a few words, new words today that I hadn't heard of before. And it was also, I think, quite nice to understand the different way that these companies are trying to work on different vaccines. And it's certainly, vaccine is not a one-size-fits-all model. I think the debate around who gets the vaccine first is going to be quite a contentious one ultimately. But we'll have to see how that ultimately plays out. I suppose ultimately it's not really in our control, so we can only hope that the people who do make those decisions make it in the absolute fairest and equitable way. Yeah, I can't see any other questions. So if there are any further questions, people are more than welcome to send me an email. But I want to thank you, Dr. Marie, and also Dr. Duma for your presentations. To everyone who's joined us, thank you for joining us at this time of the year. We know it's busy, but we wanted to at least just end the year off with some valuable information and some food for thought. I do hope that everyone spends good quality time with their loved ones this festive season and stays safe. COVID fatigue is a real thing. I think that we all need to be aware that even the tiniest slip up, even the most unintentional slip up, could put us in a position where we're at risk for contracting the disease. So I wish everyone a good festive period, and I just hope that we will all just maintain our guard, keep following those non-pharmaceutical interventions. I think we really are close to getting to a point where the pandemic is at a point where it can be controlled. But until then, we need to do our best to limit the spread of the virus. So to everyone, all the best for the festive period, and we will see you again in the new year with more interesting topics. Thank you very much.